Can I simulate your future? Yes, I can. If I understand your microbiome, and if the model understands the dynamics of the microbiome, then I can simulate all the thousands of different scenarios, right? Like, what if you eat KFC from now on forever?
I wanted to thank you for coming.
Thank you so much for having me.
Yeah, you are the leader of one of the research groups at Sano Center for Computational Medicine. Would you tell us a little bit about yourself and the group that you're leading?
Sure. So my name is Tomasz Kościółek. I'm the leader of the structural and functional genomics team. So our primary interest and object of studies is the microbiome. But we approach the microbiome slightly differently than perhaps many other research groups. So we're thinking, and the name of the group is the secret here, in terms of structural and functional genomics. So we think of the microbiome and of the problems of how to approach understanding the microbiome is through understanding the building blocks that, in effect, amount to the microbiome.
Well, I was actually, trying to understand everything that you do in your team. At the very beginning, I was a bit, must say, confused because there is lots of tools that cross biology and informatics. But in a way, it's very simple. So if life is built by proteins, then you can predict how they fold and the shape will kind of correlate with the function, and you trying to understand what it means, right?
Essentially, yes. So oftentimes, even within my group, I jokingly say that we study the microbiome because higher organisms like eukaryotes, like humans, are too complicated. And there is really truth to that, because the microbiome, so the different bacteria and other microorganisms that inhabit everything and everywhere, but most importantly for us, the human body, right? So we've got the skin microbiome, the oral microbiome, the microbiome on our palms is going to be different than the microbiome on our legs and feet. But most importantly, we've got the gut microbiome. So the microorganisms that inhabit our gastrointestinal system, most importantly, our large intestine. And there's a huge variety of them. And this is important. So microbes, unlike humans, so we have roughly 20,000 different genes within our genome. And this amounts to this incredible, diversity that we see. But it amounts through a variety of quite complex mechanisms. But when we talk about the microbiome, microbes are essentially those way smaller organisms than any human cell. And there's just a huge variety of them. So if we think about the microbiome from genes perspective, there are tens of millions of genes within our microbiome. So the orders of magnitude more, right? So we have now this opportunity to try and understand what this wealth of different genes, what they're doing actually, and how this in turn translates to the effect that we see. Because we, in fact, it's been well studied over the past decades that changes in the microbiome, which occur through, as we live our lives, as we sleep, as we move around, as we eat different things, as we change our environment.
So human body is inhabited by lots of microbes, inside and outside. You're focusing on the microbiome that is inside, in our guts. And I really like once you said in one of your lectures that it's like an organ really, right? Kind of dynamic, changing, but also it's a lot of it, like 1 kilogram or 2 kilograms?
2 to 3 kilograms, right?
Wow.
So a good analogy oftentimes that's used is the microbiome or gut microbiome is like our brain. Right. Similar in size, similar in sort of dimensions, the weight, and similar in its importance.
Right. Well, there is a saying, I can feel it in my gut, right? So, yeah, I absolutely agree that there is something more than just, a digestion process, right? It impacts other functions. So we've got that. What enabled you, what kind of other discoveries enabled you to move ahead and try to look into the microbiome functions, how it behaves? What was the most kind of helpful in that pursuit?
Yeah, so I guess one of the enabling technologies of recent years was AlphaFold, really. So our ability to predict protein three-dimensional structures from sequences. It's a little bit unintuitive, perhaps, in the beginning. So kind of let me maybe walk through this, how this kind of all amounts to being important. So essentially, we learn about the microbiome not by using a microscope or not by, using those agar plates. Instead, we're learning about the microbiome through metagenomic sequencing. So in essence, to characterize the microbiome in different proportions of different organisms or different genes, we extract all of the microorganisms, we break them down to arrive at their DNA, and then we sequence the DNA. So this is our input information. So DNA, if we go back to molecular biology and the basics of molecular biology, DNA encodes RNA, and then RNA is being translated into protein sequences, which then in turn become folded and execute whatever their role is in the organism through their shape, essentially.
Yeah, so it's biological communication, right? Not words, but all this chemical and biological structures.
Yeah. So our point of entry is this DNA information, and over the decades, we have perfected the reproducibility of DNA sequencing and the price, essentially. Right? Because in the year, at the turn of the century, one of the greatest human achievements was sequencing of the human genome. But it was a multi-billion-dollar project. And we arrived at those 20,000. We arrived at way more than those 20,000 human genes, but in terms of scale, tens of billions of dollars of cost amounted to less than a single kind of microbiome sample today. So over those 25 years, we've made tremendous progress in those technologies. So nowadays, as of 2025, 2026, acquiring the information about DNA sequences is cheap. What's expensive is everything that's kind of downstream. So people have also been very interested over decades, since the late 60s, regarding protein three-dimensional structures, right? This is our key to understand the biological role. And little by little, X-ray crystallography experiment by X-ray crystallography experiment, because this is essentially how we learn about protein structures experimentally. We have collected all those different structures, and we started, first of all, we started seeing some patterns that helped us to start making sense of all of this. But second of all, Christian Anfinsen, the Nobel Prize laureate, was pivotal in this because he proved that the protein sequence determines its three-dimensional structure. So that was a key moment later for the emergence of bioinformatics.
Right.
So people since early 1990s started attempting in a controlled environment to try and determine whether we can build algorithms that allow us to input the amino acid sequence and get a three-dimensional structure at the end of it. We were horrible at this in the early 90s. But we had the tools,
right?
So, and I'm saying about all of this because it's easy to point out at AlphaFold and say, like, this was the breakthrough. But this was a breakthrough 40 years in the making. So I think it's very important to appreciate that. So then over the years, we, as a kind of research community, we had those, first of all, methods to measure how well we're doing, and the setting to check that or to evaluate that. And that was the CASP, which is the critical assessment for the method for protein structure prediction, CASP in short. So this is, as I like to call it, kind of the protein structure prediction Olympics, but that's happening every 2 years, so that everyone gets to participate in a fair competition, because no one knows the answer, just the organizers do.
Cool.
So back in 2017, 2018, because that was happening across this time, AlphaFold 1 came along, and AlphaFold is coming out of Google's DeepMind lab, and they're going to be very important also later on. So they created AlphaFold 1, and it was a very big moment for the protein structure prediction community, but it's not the breakthrough that everyone's talking about.
Right.
Because AlphaFold 1 was excellent, Google class engineering that was building on top of those decades of incremental improvement. And they showed that if you add excellent engineering to it, you can make really good progress. But AlphaFold 2 really changed the game, because then they went back essentially to the drawing board, the researchers under John Jumper, and they redid everything from scratch, essentially. But that was possible only because there were decades upon decades of people laboriously accumulating the biological data, and decades upon decades of researchers thinking, how can we reasonably measure the progress that we're doing? So with all this in place, it was really a perfect time and opportunity, and also technology with GPUs, with deep learning, with all this progress that we're calling AI today, it made it possible.
Right.
So now, as of today, in fact, the protein three-dimensional structure, and some structural biologists are going to be very mad at me for saying that, but it's a commodity.
Sure.
Because it's easily accessible. It's not perfect by any means.
Yeah.
We still have many open questions, but importantly, we can reliably predict three-dimensional structures and know whether we're doing a good job or a bad job. And second of all, we can do this quickly.
So if I compare it to the development of transformers that are, everybody uses LLMs and everybody talks about it. This sort of is the same pattern. So huge work done by AI community, by people trying different symbolic way of coding, then machine learning, and then finally attention is what you need. We're coming the same year, 2017, right? Coming to life, and that kind of method enabling LLMs. But also in AlphaFold case, do you think that also the most important thing was that community could access it, openly, right? So it was open source, it was given, right, to research community.
Eventually.
Yeah, eventually.
After some pressure from the research community, but also, that was done for safety purposes on DeepMind and as well.
There are some other versions that are still, I think AlphaFold 4, that is not given to even research community, because there are some concerns about safety, right?
Absolutely, because the future of subsequent AlphaFold 2 was this big breakthrough. Then subsequent AlphaFold versions, DeepMind researchers also started focusing on drug development and drug design.
And that is powerful.
And that is very powerful.
AlphaFold also, not AlphaFold, but DeepMind, spin-off isomorphic lab, right?
Correct.
So that is on the top of technology to build a capacity that can be business-oriented and create money, right?
But mind you, so we're talking a lot recently about LLMs and how powerful they are, and the progress that OpenAI and Google and DeepMind are doing. But see, those pivotal technologies that the leaders of DeepMind, for example, believe in are substantially different, right? Because AlphaFold is not an LLM by any measure.
Absolutely.
Right? And the subsequent methods and the subsequent breakthroughs that are coming out of DeepMind, with Gemini, of course, but this is also a wide collection of different specialty purpose-built models. So there is equal measure of progress and development going on in those very specialized, very powerful models.
Right.
and in those general use LLM-based models. Kind of want to highlight that because in our work, the work of my group, the specialized models are very important, and this is the type of approach to deep learning, to AI that we are preferring.
Yeah. So let's go away from AlphaFold and DeepMind and Google. Let's try to focus on your tools that you created and can be applied on the top of other tools that's been released and are open source. Let's talk about DeepFry.
So DeepFry stands for Deep Functional Residue Identification, but we obviously made it post hoc because we just like the name. But it's a method to predict or to annotate protein function on the basis of its sequence and 3D structure. And we were working on this methodology together with our collaborators from the Flatiron Institute in New York. While we were working also on an effort to predict a large amount of protein 3D structures. Mind you, that was pre-AlphaFold era. So that's why I mentioned this computational efficiency, because for us, it costed us thousands of CPU hours to predict one protein, roughly 50-50, having a chance of it being a correct 3D structure, whereas AlphaFold is roughly 1 hour.
Right.
And 90 odd percent chance that you are correct. So huge change. But we were working on that problem. So we had an opportunity to predict a lot of three-dimensional structures. And we were thinking like, well, we can kind of look at all of them, but there's just going to be too many. So how do we reason about what those proteins can be doing? Well, we need some sort of an algorithm. So our thinking initially was very, very simple and dictated by this need, right? We have an opportunity, we have collaborators who can run those predictions at a scale. Well, now how do we do something useful with that? Yeah. Essentially, that was the challenge. And the answer was a direct continuation of this logic that I explained to you in the very beginning. So DNA to RNA to protein, 3D structure does things, right? So how can we, a protein 3D structure does things, can we kind of translate this into an algorithm? And it turned out that was, also very serendipitous time because of the transformer, because of the progress and the whole explosion of different methods. And we landed on a technology that's called graph convolutional networks. So we essentially changed our three-dimensional structure into a graph. And the kind of the insight here is that when you look at the sequence, you're just able to look kind of on a line, right? But when you look at the structure, then things that are not necessarily close to each other in the string sequence may be close to each other in the three-dimensional structure. Therefore, the information kind of between the two that's being exchanged may be important, right? Because this is a part of the function that they execute. And it was very efficient. It turned out to be a very, very efficient approach, which we then kind of took further. So looking at DeepFry, well, DeepFry performs really well. We're able to understand when it makes a confident prediction versus when it's not sure about what it predicts. This is important for us then using this downstream. But then at the same time, essentially, protein 3D structure became a commodity. So wonderful. So now we have all of this abundance of structural information and an algorithm that we developed kind of in parallel, not knowing that this is going to happen. So when we put the two together, all of a sudden we have the capacity and ability to annotate thousands or millions of protein structures. Well, what has millions of different proteins? Well, the microbiome. So now we're in an excellent position to start looking at the microbiome using a very different lens, if you will.
And also, I have to make this clear, when you're talking about analyzing samples, those are stool samples, right?
Yes.
And your work in New York, am I correct? Do I remember correctly that there was a national kind of research that people could, for $100, could have their microbiome tested and the samples were collected? Was that?
So that was not in New York, but in San Diego instead.
Because you did the PhD in San Diego, right?
I did my postdoc in San Diego with Rob Knight. The focus of the group was and still is the microbiome. But, it's a big group. We've been doing a variety of different projects. But then what you're mentioning is the American Gut Project.
Right.
So the American Gut Project kind of came about as a realization that after the Human Microbiome Project, that the microbiome is not as clear-cut of a question as the human genome is. Because we are 99.9% genetically identical. So kind of having one human genome covers a lot of the information that we need. Then we just need to focus on what makes us different to try and understand that. Microbiome, especially the human gut microbiome, everyone's microbiome is unique.
That's right, yeah.
So the Human Microbiome Project, the realization was we won't solve the problem with just one single project. So the continuation of this was, then we need to turn to citizen scientists.
Okay.
So for $100, you can have your microbiome characterized. You contribute to science. You couldn't at that point in time, so like 10 years ago or more, you couldn't turn it into any reasonable, actionable information, but you can have a cool piece of information about yourself.
Yes.
And you can contribute to this knowledge base of us understanding and trying to appreciate this microbial diversity better. And as far as I understand, the American Gut Project is still active, and it was also very important and very impactful in terms of inspiring other countries and other places to pick up similar initiatives. Because if the American microbiome is so diverse and so unique, then surely the British microbiome is as well, right? And it turned out that this is indeed the fact. And then the Chinese microbiome, this must be very different. Well, in fact, it is. So for us to be able now to turn our knowledge and understanding of the microbiome, which we're constantly still building, we need to understand those national baselines, if you will, whatever that means, because geography dictates the diet, dictates the climate that surrounds us, but also our own genetic diversity, our own kind of cultural background, which will impact how we live, what we do. This all has a systematic effect on the microbiome. So then, in effect, to be able to understand the relationships between the microbiome and disease, and the relationships between the microbiome and how we can improve our lives, our well-being, is very closely tied to us understanding like what's in those local environments.
We will talk about the kind of benefits of understanding gut microbiome in a second, but I wanted to focus for a while on the challenges. Yeah. So you also create standards, how to compare different samples and how to make sure that actually we collecting and measuring something that is correct. Could you tell us a little bit more about this? Why it's important to create standards?
For sure. This is very important. So thank you for this question. So the microbiome is huge and diverse, and the techniques that we're using for studying the microbiome are sensitive.
Right.
So in essence, and I think this is at the core of this, like whatever we do with the sample, considering that our technique, our sequencing is very sensitive, every step has an impact. So we need to make sure that the data we collect is reproducible, or as I prefer to put it, is wrong in the same ways, right? Because we will always introduce some sort of a variation, some sort of a bias. But as long as we understand what kind of bias we are introducing, then we are in a position to make those general comparisons, to draw those general conclusions. So this is very interesting with relation to kind of me coming back to Poland and setting up the research group. So that was back in 2019. And as I came back, I wanted to kind of integrate with the Polish research community, and then started reaching out to different people, different doctors and so on. And the interest in the microbiome was huge. I was very relieved, so to speak. And what worried me a little bit is that everyone had a different idea about how to go about collecting, storing, and sequencing those samples. Well, those were just the lessons learned across the pond, just by me.
Yeah.
So it was quite easy for me to say, stop, let's think about it. How can we make this be useful just beyond your individual project?
Right.
Because if you use a different collection device, a different sequencing platform, if you store it in a different buffer or a different liquid, essentially the sample, this will impact what you see at the end of the day. So now standardizing this, and I've been doing this work back in the States. Now I'm preaching and doing that work in Poland and in Europe, and we're working on those standards across the board, right? Because this is how we collect samples, how we sequence samples, then how we store data, and how we exchange the data.
Would you give any specific example?
Oh, sure. Well, essentially, so we characterize the gut microbiome using a stool sample. In essence and in practice, what we often encounter is we just collect those samples using swabs.
Right.
And then it even matters what kind of a swab do you use, right? So in the beginning, we were using, for example, swabs with plastic handles versus swabs with wooden handles. Seems like a trivial difference.
Okay.
Turns out that plastic swabs leave plastic residue within the sample. So if you're trying to do different experiments on the same samples, this doesn't concern metagenomics specifically, but it concerns metabolomics, for example. Whether those plastic swabs makes it unusable for metabolomics, you have to use wooden swabs. So those are tiny, minute details, one would think, that make those differences. Well, we can go further, right? So we're doing metagenomic sequencing in essence, but what is happening in practice is we break down all the cells, we chop the DNA into pieces, then we need to multiply it because in this tiny little sample there is not a lot of this DNA, so we need to multiply this DNA. So we have this huge and complicated soup of DNA coming from very different organisms, and this DNA is sometimes very repetitive and so on. So as you sequence it, you get a collection of quite short fragments, which is a computational challenge. But it is a challenge that we can circumvent if we do long-read sequencing, for example. And it is a novel technology that, introduces this, on the surface, a trivial change that we're going from fragments of roughly 100 to a few hundred nucleotides, and now we have fragments of a few thousand nucleotides. It makes it infinitely easier now to go back from this alphabet soup back into sentences that you have those much longer fragments. And this is the kind of technology that I'm kind of advocating for and pushing, because it made our analyses way more robust and reproducible, and it made a lot of the downstream challenges that we as bioinformaticians are dealing with much more tractable.
Okay, so the gut microbiome that you also work was not from adults. There was also gut microbiome from infants, from babies, right?
Yes.
That's also
absolutely
interesting.
That's also fascinating, right? And this is a very different world. Because the microbiome and as we live our lives, the microbiome matures with us.
Right.
And the first microbiome is the microbiome that we get from our mother.
Yes.
So now the mode of delivery, how we arrive in this world, whether we are born through the forces of nature or through a C-section, it makes all the difference in those early days. It's still a matter of scientific debate whether it impacts your life long term But for sure, we do see that there is a difference in the microbiome of babies born or infants born
naturally
versus C-section. But also this microbiome, as we mature and grow up and become teenagers, so we are all
start drinking,
the hormonal changes that impact us, but also our diet changes, right? We start with this baby diet, which kind of feeds different microorganisms. Then as we turn to more adult foods, and so on. So it's all very fascinating, and that's also a big challenge, I would say, because on one hand, our microbiome is our own. So the microbiome that we have at the age of 18 or 20, like it's our top microbiome that we might want to preserve, for example, right? So we also started thinking about, for example, biobanking initiatives, because we're noticing that the microbiome is highly personalized. So we figure like, okay, can we actually store your microbiome as you are in your prime and then use it when you need it, right?
When you need to rebuild it.
When you need to rebuild it, exactly. So antibiotics, right? Obviously this is a wonderful and cornerstone of our medicine, but it's a double-edged sword, right? Sure. It destroys our microbiomes, and oftentimes, with severe or significant diseases, restoring this microbiome after some aggressive course of antibiotics is a challenge.
So now we going in our conversation to translating this discovery into something that is actually impactful on the level of a patient, of a human being, right? Yes. You don't have to be sick. You can be, you can get better through the microbiome. Your well-being can be. You don't have to wait till the symptoms of a disease. So you were talking about the about antibiotics, but there is also something very interesting for me in your work, which is gut and brain axis, right? And that's actually. It can be very impactful, still need lots of research, but would you tell us a little bit more about it?
Sure, absolutely. So I started by comparing a little bit the gut to the brain because of its role and size and scope, but in fact, there is also this connection between the gut and the brain. We have several levels by which this communication can occur, but first maybe let's kind of build an intuition, right? So if you're stressed out, you oftentimes don't feel like eating, or you may sometimes feel like, nibbling a little bit to calm your nerves. Well, this is gut-brain communication right there. This is a living example. So we have this direct communication. So what's happening in our brain can impact, how our guts move. Well, this will impact the structure of the microbiome. There is a vagus nerve communication also going on both ways. So in principle, the microbiome can activate vagal communication, and the vast majority of neurotransmitters are produced in the gut.
Yeah.
Hormones are produced in the gut. Inflammation is being regulated and often comes from the gut. So there is this wide-ranging, the both kind of direct and system level communication between the brain and the gut, and we do see it manifesting across the board, essentially. So for people with depression or anxiety, we do see that their microbiomes have certain different signatures. For people with severe mental illness, with schizophrenia, for example, I did several papers studying people with schizophrenia. Of course, they're medicated people. They're people who live very different lives oftentimes, but we're trying to also understand and, disentangle all of that to try and help. But also, this gut-brain communication may have an effect in neurodegeneration, in Parkinson's, in Alzheimer's disease. We do see, because, those microbes that inhabit our guts don't just live freely. they often form communities, different microbial cities, if you will, and they're building those cities. They're using amyloids oftentimes to anchor themselves to the lumen of the gut. But those same amyloids that are being produced in the gut are very, very similar to those amyloid plaques that are associated with the progression and development of Parkinson's disease.
You know, in Poland, there is this, how do they call it, mieszanka studencka, student mix, right?
Yeah.
The raisins and nuts,
Trail
basically,
mix.
right?
Trail mix.
To kind of, for people who are going to use their brain for studying, right? Have a snack like this, graze on something that will be a food for your brain. So that's obviously, you know, silly example, but you're taking it more seriously, right? And so now we've got the big impact of your research can be personalized medicine, can be new ways of seeing if the person is indicating some kind of conditions that may trigger neuro diseases. So there possibly will be biomarkers, new biomarkers, right? And also restoring the well-being, restoring kind of, or maybe not restoring, but just like prolonging health, right?
Essentially, yeah. So there are two things I'd like to say here. So first of all, Sano Center for Personalized Computational Medicine. So exactly, you know, this is our mission. But yeah, I guess obviously we're also thinking about health in a less binary fashion nowadays, right? Health is sort of a spectrum. We may not necessarily have a certain disease, but we may be heading there. We may have no diseases, but we may not still be in perfect health. So thinking more in those well-being terms, I think this is a very good direction. So for sure, as the future of personalized medicine, I see the microbiome like this is just, to me, just such an obvious component, right? The technology is almost there, right? It's getting cheaper and cheaper, more accessible, more reproducible. I think it's just a matter of time until either everyone does their microbiome characterization at home. We have a smart toilet, you know?
Yeah.
You just
Cool idea.
You just do your business in the morning, and you know, a few moments or a few hours later, you get a notification on your smartphone, like this is what you need to eat today. This is what you need to
drink,
because this is we can measure that, you know. Because otherwise, you know, if you don't do this, your risk of neurodegenerative disease in the next 10 years increases by, you know, X%.
You're also entrepreneurs, so you're actually doing on the top of your research, you know, all these things that can be useful for individuals, right? So would you tell us a little bit more about one biome?
Yes, one biome. Yeah, that's the name. So exactly, Kasia, as I said, I don't think we have the personalized medicine just yet. But what we can do with the microbiome already is characterize it and figure out what types of microbes we have too much, what types of microbes we have too few, and then how to modulate that in this one health perspective, right? So health is not just a lack of the disease. Health is something we can, you know, optimize. So one biome is doing exactly that. You know, we get a sample from a client, we sequence it, characterize what's in the sample, and using our knowledge coming from scientific literature about microbiome and dietary products, how they, you know, interact and how we can modulate it. We're using AI technologies to then come up with a set of recommendations of how to improve and optimize your microbiome. But then we also include, because those are, first of all, early days, second of all, human in the loop. That's always a good practice. So those results go to a qualified dietitian, who then in consultation with you comes up with dietary and lifestyle and supplementation recommendations that can help improve your microbiome specifically.
So I heard the feedback from one person who was going through the procedure in your company. So he said that he's not allergic to gluten, but it occurred that he ate too much bread products, like wheat products. And the advice was that he drinks, before he starts eating breakfast, he drinks a glass of cold water with 1 spoon of apple vinegar, right? Yeah. That's the trick. I mean, that's so easy, right?
Sometimes there are very simple solutions that have a very profound effect. Sure. And for some people, it just really works that we can just measure it. And we can, hold them accountable to a certain extent, And this is also, the microbiome is also a fascinating kind of investigative tool to a certain extent. So we had another client, and I was taking part and taking notes during the consultation as we were, improving and developing our product. And dietitian that was leading the consultation, she asked the client, well, I would recommend you cut down on smoking. And the client responded, but I don't smoke. I was like, oh my God, wow. What did we do? We made a mistake. But then the dietitian was like, no, you've got all those microbes and this comes out of AI. I'm like, oh my goodness, no, we're in trouble. But then the client reflected no, I take snus.
What's that?
The tobacco pouches.
Ah. And it's visible in the gut.
It's visible in the microbiome. So we wear those, the next, we see that you're sucking. You don't lie to us.
Excellent. Okay.
So there is this whole, variety, because also there is a matter of either personalized or more targeted probiotics. A lot of the probiotics that are developed and that are available on the Polish market, from what we see, are not necessarily suitable for Polish population. And there's a good reason for that. We're buying the strains that are have been developed internationally usually, and we're just copy paste them into the Polish context. But out of the clients that we've handled so far, for example, we're seeing that we have absolutely no problems with the abundance of lactobacilli.
Right.
Our Polish diet involves a lot of, fermented foods, a lot of milk products, and so on. But for example, we see that Polish people have very few bifidobacteria. Mm-hmm. But there is no, the general need and the general probiotic mixes include both lactobacillus and bifidobacteria. Mm-hmm. But they're developed for the more international population. What we do see is that possibly, and I don't want to say this too strongly yet, but we do see that we do have our own unique needs in Poland, and it just is very likely that one of the future scenarios is that we are going to have either those very specialized probiotics, but specifically for you, or for different types of subpopulations, if you will.
Excellent.
So this is all very exciting, right? Because we have this hard kind of medicine direction where we can diagnose diseases on the basis of the microbiome, or at least, indicate risk. We have this kind of middle ground when we can play, detectives and figure out the different things that, we do and are not necessarily good for us. But we also have this direction of just impacting our well-being by just those subtle changes, water with apple cider vinegar in the morning.
Absolutely, it makes a difference. So I really think that your work strongly connects to Sano's vision to create digital twins that will be able to deliver that promise of personalized medicine. How do you see this?
Yeah, the digital twin technology has emerged from this modeling and simulation community, right? Let's think about the human body as this physical challenge in physics of simulating the flow of blood and the bone structure and so on. And now as we're moving further and further with the technology and with our understanding and appreciation of the microbiome and the general technological progress around AI, machine learning in more general terms, I see that there is a huge opportunity not for those mechanistic digital twins, but for the AI-based digital twins. And this is one of our directions and ideas that we want to follow up on. So, of course, there is the huge impact and influence on human health, and there's the huge variety and variability within the microbiome. So we do see very much inspired by Google DeepMind's approach. Let's build an expert system by us understanding all of the nitty-gritty steps of this problem. So the way that we're approaching building the microbiome digital twins is to have a foundation model of sorts. So a model that understands the structure and the dynamics of the microbiome. Or, as I like to put it sometimes, if we think about ChatGPT or Gemini and all the different LLM-based models, well, they're essentially models that understand human language. I mean, this is what they were trained for ultimately. The intelligence kind of emerges as a byproduct of the model understanding the language. So taking a very similar, in principle, approach is what we want to do. If the model learns from the data how the microbiomes of different people look, how the microbiomes of different people change, it will eventually understand the language of the microbiome, the language of different microbes and different pathways, different functions that they are capable of executing. And we already have that. This is still preliminary, but surprisingly, this model understands the microbiome really well. So now we're turning this model that can, speak the language of microbiome. What we are planning on doing in the near future is actually start fine-tuning it to be able to understand an individual. And this is still an open question. Like, just do we need one sample from you to understand your microbiome? Or maybe we need 2 or 3. But once we are able to do that, we're able to achieve all of the objectives really that the digital twin has, right? Can I simulate your future? Oh, yes, I can. If I understand your microbiome, and if the model understands the dynamics of the microbiome, then I can simulate all the thousands of different scenarios, right? Like, what if you eat KFC from now on forever,
I can imagine what would happen. Yeah.
And so on and so forth, right? So this construction of the digital twin for the microbiome, I think, is now becoming possible because of the hardware and software that we've got. So the kind of the computer science, computational solutions, because the data is becoming abundant and reproducible enough. So this is, a lot of the work that I've been putting into this, right? We need to be able to learn true biology and not all the different quirks of different machines and so on. And it's become accessible, it's become financially accessible enough. I mean, maybe we're not there yet to do this for every citizen of Poland.
Sure.
But we're able and will be probably able to do this in all the situations that weren't such an characterization. And this is becoming, it's happening as we speak, essentially.
Exciting. I'm looking forward to the digital twin of microbiome. Thank you very much, Tomasz.
Thank you.
Thank you. That was great.